Hello, I'm Dr. Hannah Roberts. This presentation will provide an overview of how Glycoscience can shape the future of pharmaceuticals and personalised medicines. The number of therapeutics based on glycans, glycan targets, and glycosylated products is rapidly increasing, and they are an important group of biopharmaceuticals. In 2010, approximately 40% of all approved therapeutic proteins and 8 of the top 10 selling biologics, meaning it was manufactured in a living system, contained N-linked or nitrogen-linked glycans, and it also generated over $35 billion in annual sales. Glycosylation is the addition of glycans to a protein. The surface of all cells is coated with a layer of glycans called the glycocalyx. One of the major challenges is to find ways to understand glycosylation of biologics to create a defined targeted glycan profile. There is a particular demand for tools that can address this challenge, which could lead to better in vivo product characteristics, increased biotherapeutic efficacy and safety, also quicker routes to markets, better tools for glycan profiling would also be a great benefit to biotech, pharma and biosimilar industries, instrument manufacturers, as well as the healthcare sector. Patients will also benefit through the development of biopharmaceuticals that are lower cost, safer, and more effective. We will now look at a specific case study. Perhaps one of the most well-known examples of a biopharmaceutical is erythropoietin, known as EPO. It's a glycoprotein hormone that controls red blood cell production. Approximately 40% of its weight is due to glycosylation. In the picture, the glycans are shown in red linked to the protein in blue via individual amino acids, which are orange. Advances in recombinant DNA technology have allowed the production of recombinant EPO for therapeutic use in treating anemia. First approved by the FDA in 1989. Glycoscience has played a major role in improved EPO variants, with modified glycosylation bringing them to the market. One such product is Aranesp from Amgen, and it has been found to have significantly longer half-life in the body after injection, providing improved therapy. It is anticipated that worldwide sales of EPO will continue to grow as even more effective variants for anemia treatments are found. All cells, including human cells, have carbohydrates on their surface, known as the glycocalyx. This coating of glycolipids and glycoproteins controls a variety of fundamental biological processes, for example, helping the sperm to recognize eggs during fertilization, determining blood groups or enabling the body to identify disease cells or infectious agents. The carbohydrates of the glycocalyx are produced through highly complex biosynthetic pathways, controlled by genes and the environment. The makeup of the glycocalyx is therefore highly sensitive to genetic mutations, changes in gene activation or silencing, or environmental factors, such as diet, alcohol consumption, or smoking. This makes the glycocalyx a useful target for personalised medicine, including finding new biomarkers for diseases, such as cancer, or for patient stratification or subgrouping in clinical trials. Other opportunities which are of great interest include developing new vaccines, antimicrobials, anti-cancer drugs or diabetes treatments, and creating more effective and cost-effective production processes. By building on and underpinning the recent success of biopharmaceuticals, as well as reducing healthcare costs by using carbohydrate-based biomarkers to develop more effective disease diagnoses and better targeted medicines. For example, many viruses attach to the human cell surface sugars. A tool called carbohydrate microarrays can be used to understand this process and have uncovered how seasonal, pandemic, 2009 swine flu, and bird flu viruses differ in their cell surface recognition. In 2009, the pandemic of H1N1 influenza caused thousands of deaths worldwide. but the majority of cases were relatively mild. A variant of the virus carried the mutation termed D22G in a protein on the surface of the virus, and people infected with this variant were more likely to have severe and fatal illness, as the mutant virus could have impaired the lungs' ability to clear out germs, highlighting the potential for deadlier strains of flu to emerge and spread. As personalised medicines advances, there will be many opportunities to develop lower cost and better targeted products for the diagnostics market, such as on-in-body sensors, nanotechnology-based sensors for use in the field, fast effective diagnostic tools for the detection of pathogens, and monitoring of health and disease will greatly benefit society, both reducing human suffering and cutting healthcare costs. These tools will also allow governments and healthcare providers to put preventative measures in place, for example vaccination programs in infectious disease outbreaks, or targeted inventions for diseases such as cancer and diabetes. These new opportunities will be a great benefit to the pharma, biotech, biosimilar industries, diagnostic companies, and to the healthcare sector. The glycocalyx can be mimicked in the laboratory using carbohydrate arrays, where glycans are displayed on the surface of microarrays. Such glycoarrays have been used to identify biomolecular interactions involved in signaling on cell surfaces. Glycobiomarkers have enormous potential for use in diagnostics and personalised medicine. High-throughput screening technologies, such as gold glycan arrays, have been used to identify new glycan biomarkers for a range of diseases. For example, during a 5-year FP7-funded project, the tools for the detection of novel glycobiomarkers, or glycobiome for short, a variety of different biomarkers have been discovered and are now under commercial assay development. We have maturity onset diabetes of the young, or MODY, colorectal cancer, Crimea-Congo fever virus, herpes simplex virus, and CA125 biomarker for primary invasive epithelial ovarian cancer, and we can detect between benign and cancerous tumours. European scientists have carried out significant research into the role of glycans in stem cells and the application of glycan-based technology for regenerative medicine and tissue engineering, such as materials for organ replacement and patient-specific testing of therapeutics. This image shows bone marrow-derived stem cells. Cell nuclei are in blue, actin cytoskeleton in red, and the sugars are in green. Stem cells, known as a characteristic pattern of sugars, shown here in green, direct the behaviour and can be used to target them in vivo. Glycans on the surface of cells reorganise as cells differentiate, producing tissue and cell type specific glycans, which determine cell fate. Glycans have also been found to be key molecules that direct cell behaviour, making them excellent markers of stem cells, offering attractive targets for the specific delivery of therapeutics or toxins for cancer treatments, or as tools to manipulate stem cell differentiation in vitro. Stem cell therapies also have the potential for treating congenital diseases of glycosylation, or hereditary diseases with impaired glycosylation processes, some of which are lethal. To summarise the opportunities within pharmaceuticals and personalised medicines, we have the development of new vaccines, antimicrobials, anti-cancer drugs and diabetes treatments to create more efficient and cost-effective production processes by building on the underpinning and recent successes in biopharmaceuticals. Also, reducing healthcare costs by using carbohydrate-based biomarkers to deliver more effective disease diagnoses and better targeted or personalised medicines. We have here some excellent links to Glycopedia and IBCarb YouTube videos, which provide a little bit further information.